Okay, so welcome back to this next video in which we are discussing asthma and anti-asthmatic drugs. Okay, so we're in the process of discussing the pathway downstream of activated Fc-epsilon-R1 receptors, and specifically we want to see how that pathway activates the production of cysteinyl leukotrienes. So we've seen that downstream of the Fc-epsilon-R1, which is activated, you're going to get the activation of the Ras-Raf-Mek-ERK cascade, and this enzyme ERK, also called MAP kinase, is going to phosphorylate the enzyme phospholipase A2, and we're just about to discuss what phospholipase A2 is actually going to do. So its substrate is again a phospholipid within the phospholipid bilayer, but its substrate, unlike PIP2, is one of the main phospholipids that makes up the phospholipid bilayer, which is this phosphatidylcholine, also called lecithin. Okay, right, so let me now show you the structure of lecithin, and then we'll discuss what phospholipase A2 is actually going to do to the lecithin. Okay, so phosphatidylcholine then. As the name suggests, the phosphatidyl means that the molecule is based on phosphatidic acid, and then you're going to bind choline onto the phosphatidic acid molecule. Okay, so let's have a look at this structure of phosphatidylcholine then. Okay, so here is the membrane here, okay, and we'll start by drawing phosphatidic acid here. Okay, so here are the long-chain carboxylic acids attached to the glycerol molecule, and here is the phosphate group coming off the alcohol group off the third carbon of the glycerol molecule. Okay, and now to convert phosphatidic acid into phosphatidylcholine, what we now want to add on is we want to add on a choline molecule onto the phosphate group of the phosphatidic acid, like so. Okay, I'm going to represent the choline molecule by this rectangle here, which I'll colour in blue. So let's now colour in all of the different portions here. Okay, so in blue, this blue rectangle is the choline molecule. Okay, this blob here in purple is the phosphate group. Okay, in orange here, these are the long-chain carboxylic acids. Okay, and they're esterified onto the first and second carbon's alcohol groups of the glycerol molecule. Okay, so this is a picture of phosphatidylcholine, or lecithin. Okay, and I'll stress again that this blue rectangle here, this is representing the molecule choline, and that's why this is called phosphatidylcholine, because what we have literally done is attached onto a phosphatidic acid molecule, a choline molecule. Okay, now this choline molecule is the same choline that's present within acetylcholine. Okay, now let me just show you the structure of choline. Choline has an alcohol group, and it's this alcohol group that's going to be involved in binding to the phosphate group of the phosphatidic acid molecule. Okay, then it has an ethylene group following that alcohol group. Okay, so two carbons with hydrogens coming off them here. Okay, and then off the other side you've got a nitrogen atom, which is going to have three methyl groups coming off it here, 1, 2, and 3. Okay, and this nitrogen ends up having a positive charge, because nitrogen's not supposed to have 3 bonds coming off it. It's only supposed to, nitrogen's not supposed to have 4 bonds coming off it. It's only supposed to have 3 bonds. Okay, so one of these bonds is going to actually be where the nitrogen is giving both electrons into the bond. Okay, so usually what should happen is nitrogen should have 3 bonds, and then it should have 1 lone pair of electrons. One of these bonds, and let's say it's this one here, will consist of 2 electrons that have come from the nitrogen, and then there will be no electrons that have come from the carbon. Okay, now because the normal understanding of a covalent bond is that 1 electron has come from each member of the bond, it's basically as though the nitrogen has now given away an electron to the carbon, okay, and that's what gives the nitrogen a positive charge, because it's effectively given away an electron to the carbon. So you can then ask, well, why doesn't the carbon have a negative charge? Well, it's because when the carbon entered this sordid arrangement that we've got here, it will come in with a positive charge, and then when it received this electron from the nitrogen, it will become neutralized, and it's effectively deferred that positive charge onto the nitrogen. So choline as a molecule is positively charged, basically. It's got a whole positive charge on it. So this is our choline molecule here. Okay, so basically you take one of these molecules and attach it onto a phosphatidic acid molecule, and that creates you phosphatidylcholine, or PC, also called lecithin, which is one of the major phospholipids within the phospholipid bilayers, okay, and particularly in the plasma membrane. Right, okay, so phospholipase A2 enzymes, now that they are activated, are going to break down phosphatidylcholine molecules in the phospholipid bilayer. Okay, now how are they going to cut these phosphatidylcholine molecules? Well, they're going to cut this bond here between that second long-chain carboxylic acid and the alcohol group that comes off the second carbon of the glycerol molecule. Okay, they're going to hydrolyze that ester link, and the products then that you will produce are a phosphatidylcholine molecule which has lost that middle long-chain carboxylic acid, okay, as shown here. So if I color this in, here is one long-chain carboxylic acid attached to the alcohol group that comes off the first carbon of the glycerol molecule. Okay, there's the glycerol molecule. It still has the phosphate group coming off the alcohol group of the third carbon, okay, and then it will have the choline molecule coming off that, but it has lost that middle long-chain carboxylic acid which comes off the alcohol group off the second carbon. And this molecule that we've got here, this is known as a lysophosphatidylcholine, or lyso-PC for short, okay, also called lyso-lecithin, okay? So lyso-lecithin, lysophosphatidylcholine, whatever you want to call it. Okay, then the other product that you get will be the long-chain carboxylic acid, okay, which is here. Now, phosphatidylcholine molecules can have many different long-chain carboxylic acids in this position, okay? I never told you which specific long-chain carboxylic acid you have here, and that's because there isn't a specific long-chain carboxylic acid that you have there. There are loads of different long-chain carboxylic acids that you can have there. Okay, so when the phosphatidylcholine is broken down by phospholipase A2, you're actually going to get many different long-chain carboxylic acids being liberated now. Okay, now the one that we are particularly interested in is a long-chain carboxylic acid called arachidonic acid. Okay, and arachidonic acid has another name. It's also called icosahexaenoic acid. Okay, so icosa, which means 20, tetraenoic acid. Okay, so this is a 20-carbon carboxylic acid where you have 4 of the bonds between the carbon atoms as double bonds. So that's what the tetrene means. It means that 4 of the bonds are double bonds. So icosahexaenoic acid is a long-chain carboxylic acid with 20 carbons and 4 double bonds along that length. Okay, right. If you want the specific positions of those double bonds, it is 5, 8, 11, 14. So you might see icosahexaenoic acid written slightly more strictly as 5, 8, 11, 14 icosahexaenoic acid. But for our purposes, we'll just abbreviate it down to icosahexaenoic acid. And you can abbreviate icosahexaenoic acid even further to ETA, like so. Okay, right, but strictly speaking, arachidonic acid should be called 5, 8, 11, 14 icosahexaenoic acid to tell you the positions of those 4 double bonds of this 20-carbon carboxylic acid molecule. Okay, right, so when you break down loads of these phosphatidylcholine molecules and release long-chain carboxylic acids, occasionally it will just so happen that you break down a phosphatidylcholine molecule that has in that second position there an arachidonic acid molecule esterified to the alcohol group at the second carbon of the glycerol molecule. Okay, and when you break down that, you will now get arachidonic acid molecules liberated into the phospholipid bilayer. Okay, these arachidonic acid molecules can now be used as the starting point to make the cysteinyl leukotrienes. Okay, so let me now explain cysteinyl leukotriene synthesis from the starting point of arachidonic acid. Okay, right. So for short, I will abbreviate arachidonic acid to AA. Okay, so the first thing that happens in the synthesis of cysteine leukotrienes in these mast cells is going to be that arachidonic acid is firstly converted into something that's called 5-HPETE. Okay, so 5-HPETE. Okay, and what does this stand for? This stands for 5-hydro peroxide. Okay, so the H is for hydro. Okay, the P is for peroxide, and then the ETE is for icosahexaenoic acid. Okay, so icosahexaenoic acid, it's a long name. Okay, so the E is for icosa, the T is for tetra, and then the E is for enoic. Okay, so 5-hydroperoxy icosahexaenoic acid. Okay, so clearly that's going to be very much so related to the icosahexaenoic acid molecule of arachidonic acid. All you're going to add on is a hydroperoxy group onto the 5th carbon of the arachidonic acid molecule. Okay, now, this first conversion then is catalyzed by a very important enzyme, and this is the only enzyme in this pathway that I'm actually going to give you the name of, okay, because it's the only one that at present we actually have drugs which can block, okay, and which are being considered for use in asthma. They're not currently used for asthma, but I'll mention them when we come on to our future treatments for asthma. Okay, so the enzyme which converts arachidonic acid into 5-hydroperoxy icosahexaenoic acid is called 5-LOX. Okay, and this stands for 5-lipoxygenase. Okay, so the L is for lipo, and then it's lipoxygenase. Okay, so 5-lipoxygenase is what 5-LOX stands for. Okay, right, and as I say, there is a drug that can block 5-lipoxygenase, which I will come back to when we discuss future asthma treatments, but for now know that this drug is called Xoluton. Okay, so there is a drug called Xoluton which is capable of inhibiting the 5-lipoxygenase enzyme, and therefore is capable of inhibiting the production of leukotrienes. Now, at present, you can't appreciate yet how important that is in the pathogenesis of asthma and the pathogenesis of asthmatic attacks. Okay, so we will come back later once we've finished the entire pathogenesis of asthma, and we will re-look at this drug and why it's useful in treating asthma. Okay, but it's not at present actually used. Okay, it's one of the future treatments that might in future actually be approved for clinical use. Right, so arachidonic acid is first converted into this molecule 5-hydroperoxy icosahexaenoic acid, and from there it's now converted into a molecule called leukotriene A4, and this is all happening within our mast cell at present. Okay, so this LTA4 here stands for leuco, that's the L, and then triene, that's the T, and it's leukotriene A4. Okay, so 5-HPETE is converted to leukotriene A4, okay, and now leukotriene A4 can be converted further into other leukotrienes that are actually efficacious, okay, so it can be converted into a molecule called leukotriene B4, but we're not interested in leukotriene B4. It's not what you would call a cysteine leukotriene. We are interested in the cysteine leukotrienes, which are the main ones that are important in asthma, okay. Leukotriene A4 then is going to be converted down to leukotriene C4 inside the mast cell, and this is the first cysteine leukotriene that we are seeing. Okay, leukotriene C4 can then be secreted out from the mast cell. So let me just show this here. Okay, so if this is our mast cell here, it can convert arachidonic acid into leukotriene C4, and then the leukotriene C4, and I've missed out the L there, leukotriene C4 can be excreted from the cell, and then extracellularly it can now be converted further into leukotriene D4, and then also into leukotriene E4. Okay, so leukotriene C4 is synthesized by the pathway that I've shown you inside the mast cell. Okay, and I'll just add a nucleus on for our mast cell here. Okay, so this is our mast cell. So leukotriene C4 can be synthesized by the mast cell, and then it's released into the extracellular fluid here. Okay, and then in the extracellular fluid, leukotriene C4 can be converted further into the leukotriene D4 and the leukotriene E4. And these three leukotrienes here, leukotriene C4, leukotriene D4, and leukotriene E4, these are all called the cysternal leukotrienes, and these are all capable of going to the bronchial smooth muscle cells and causing contraction of the bronchial smooth muscle cells, as we'll see in just a moment. Okay, so from now on, we will just call leukotriene C4, D4, and E4 collectively as the cysternal leukotrienes, because all of them are being produced by these mast cells. Being produced because of the mast cells, of course. The mast cells actually only produce the leukotriene C4, but that's the starting point for the other two. Okay, and they're all going to be diffusing off to the bronchial smooth muscle cells and causing contraction of the bronchial smooth muscle cells. Okay, so to summarize again, when the mast cell gets activated, you're going to start liberating arachidonic acid molecules from phosphatidylcholine molecules. Okay? Then that arachidonic acid can be acted upon by 5-lipoxygenase to create 5-hydro peroxide cosatetrinuric acid, and this is still all occurring inside the mast cell at the moment. Then 5-hydro peroxide cosatetrinuric acid can be converted into leukotriene A4, and then leukotriene A4 can go down two routes, leukotriene B4, which we're ignoring, okay, or leukotriene C4, and that's the one that's important to us. Leukotriene C4 is what's going to be synthesized by these mast cells and then secreted by the mast cells, okay, and then extracellularly, when leukotriene C4 is in the extracellular fluid, it can then be converted further into leukotriene D4 and leukotriene E4, and all three of these are capable of acting on bronchial smooth muscle cells. Okay, so they're all now going to go off and act on bronchial smooth muscle cells. Okay, so it's not the case that all leukotriene C4 molecules that you're going to be secreting are going to be converted into leukotriene E4. Some of them will be converted into leukotriene E4, some of them will end up being leukotriene D4 molecules. You will end up with a mixture, basically, of leukotriene C4, leukotriene D4, and leukotriene E4, and that's why we're going to collectively know them as the cysternal leukotrienes, because effectively they're all going to go off and do the same thing. Okay, right. So now to summarize all of this, basically all of that great big pathway that we've now looked at can be summarized to this. Here is our mast cell with its nucleus here. Okay. Here is our Fc epsilon R1 receptor, okay, now demoted to being represented as a blob here, so that's the Fc epsilon R1, and I'll just colour Fc epsilon R1 here in pink, okay. Mounted on the Fc epsilon R1, of course, we'll have the IgE molecule, which I'll put here, which will be directed against some epitope of our allergen, and now we've inhaled our allergen. It's activated the Fc epsilon R1 receptors by binding to the IgE that's mounted on them. Okay, so here in vivid purple, this is the allergen here. And what that has now resulted in is these mast cells releasing histamine, so histamine granule release, okay, and also the cysternal leukotrienes. Okay, and these are now the two key mediators of the asthmatic attack. These are going to cause both the immediate phase of the asthmatic attack and the late phase. Okay, right. So, again, I want to stress that it's not just allergens that can cause mast cells to release the histamine and the cysternal leukotrienes. It could also be those non-allergic stimuli, such as cold air or noxious gases, so I'll put those here as well. So also cold air could stimulate the mast cells to release these molecules, and also noxious gases such as sulfur dioxide and ozone. Okay, right. So now what we want to see is how is histamine and the cysternal leukotrienes going to cause the asthmatic attack. Okay, so we firstly want to look at the immediate phase of an asthmatic attack. Okay, now the most striking part of the immediate phase of an asthmatic attack is that the bronchial smooth muscle cells are going to start contracting, and that's going to cause bronchoconstriction. Okay, so we now want to talk about how histamine and cysteinyl leukotrienes are going to cause contraction of bronchial smooth muscle cells and hence bronchoconstriction. Okay, right, so if I get back my picture of the bronchial wall, we are getting the production of the histamine and the cysteinyl leukotrienes in the lamina propria here by the mast cells, and now they're going to diffuse back to the bronchial smooth muscle cells here, and they're going to cause the contraction of those bronchial smooth muscle cells. Okay, right. So we want to see what receptors to the bronchial smooth muscle cells have on their surface that are going to respond to the histamine and the cysteinyl leukotrienes. Okay, right. Well, both the receptors for histamine and cysteinyl leukotrienes are G protein coupled receptors, and thankfully they both work through the exact same cascade, which is the GQ/11 cascade. So we're now going to just discuss the GQ/11 cascade briefly. Okay, right, so firstly let me tell you the name of the G protein coupled receptors which histamine and cysteinyl leukotrienes act on the surface of the bronchial smooth muscle cells. So histamine acts on what's known as the H1 receptor, or the histamine 1 receptor, which is a G protein coupled receptor that's GQ coupled. The cysteinyl leukotrienes work on a receptor that is called the cysteinyl leukotriene receptor, so cis for cysteinyl, LT for leukotriene, and it's the type 1 cysteinyl leukotriene receptor, so it's called cis-LT1. Okay, so we now want to discuss the pathway downstream of both of these receptors, and what's absolutely lovely is that it's the same pathway in both of them. Okay, right, so we now want to talk about bronchoconstriction, and I should just stress that bronchoconstriction is not the only part of the immediate phase. There is also the inflammatory response. However, the bronchoconstriction is the key sort of canonical feature. So we'll come back to the inflammatory response after bronchoconstriction. Okay, right. So bronchoconstriction is going to occur very quickly following the generation of this histamine level in the extracellular fluid and the cysteinyl leukotrienes in the extracellular fluid. Okay, right, and this is the hallmark feature of an asthmatic attack. So how does histamine binding to the H1 receptor and cysteinyl leukotrienes binding to the cysteinyl leukotriene receptor on the surface of these bronchial smooth muscle cells cause the contraction of the bronchial smooth muscle cells? Well, as I say, the H1 receptor and the cysteinyl leukotriene type 1 receptor are both G protein coupled receptors. So they both have this characteristic 7 membrane spanning alpha helical structure. This is the extracellular side, this is the cytoplasmic side, the N-terminus is extracellularly, the C-terminus is intracellularly. Okay, and this is either the histamine 1 receptor or the cysteinyl leukotriene 1 receptor here. Okay, right. So basically, when the ligand binds, which is either histamine or a cysteinyl leukotriene, what it will cause is it will cause a conformational change in our receptor here, okay? And then what's going to happen is that receptor is now going to be able to activate heterotrimeric G proteins. So we haven't seen any heterotrimeric G proteins so far in this video, so I'll just do a little sort of introductory speech about heterotrimeric G proteins. So we have seen monomeric G proteins. We saw the RAS monomeric G proteins. Heterotrimeric G proteins are not quite as simple as the monomeric G proteins, but they follow the same basic principle. Okay, i.e. that on-off principle. Okay, however, the complication is that they are not a single protein. Instead, they are a trimeric complex. Okay, they consist of 3 separate subunits. Okay, and these 3 separate subunits are called the alpha subunit, shown here, and then you also have the beta and the gamma subunit. So this is representing the alpha subunit, this is representing the beta subunit, and this is representing the gamma subunit here. Okay, and I'll just colour these in. So we'll have the alpha subunit of the heterotrimeric G protein here in red. Okay, we'll have the beta subunit of the heterotrimeric G protein here in blue, and we'll have the gamma subunit of the heterotrimeric G protein here in green. Okay, right. Now, heterotrimeric G proteins, like the monomeric G proteins, can be in two states. They can be in the off state, where the alpha subunit has guanosine diphosphate bound to it. So the alpha subunit is analogous to the monomeric G protein. It's the one with the guanine nucleotide bound to it. Now, when the alpha subunit has guanosine diphosphate bound to it, it associates with the beta and the gamma subunit. Now, the beta and the gamma subunit are always bound together. Okay, they never fall apart. So they are often referred to as the beta-gamma complex. So we would say that the alpha subunit is associated with the beta-gamma complex when it's in the off state and has guanosine diphosphate bound to it. Now, just like the monomeric RAS G proteins, okay, the heterotrimeric G proteins are also not integral membrane proteins. However, they are attached to the underside of the plasma membrane, and it's again through lipid moieties. Now, the alpha subunit has a lipid moiety attached to it, shown here, which anchors it to the underside of the plasma membrane, and so does the gamma subunit, which also anchors it at the underside of the plasma membrane. Okay? Now, that's the off state then. The other state is the on state. Now, the off state is the state where it's not doing anything. It doesn't signal to anything when it's in the off state. The on state, just like in the case of the monomeric G proteins, is the state where it does signal to things. Now, in the on state, the alpha subunit has guanosine triphosphate, GTP, bound to it, rather than guanosine diphosphate. Okay? And also, when the alpha subunit has that GTP bound to it, it has undergone a conformational change that means that it can no longer bind to the beta-gamma complex. So the alpha subunit will be separate from the beta-gamma complex when the alpha subunit's in the on state and has guanosine triphosphate bound to it. Okay, right. So now, these these G protein coupled receptors, okay, so this is the GPCR here, these are effectively the GEFs, the guanine nucleotide exchange factors for the heterotrimeric G proteins. No one ever calls them that, but that's effectively what they are. Okay? Now, basically, when ligand binds to G protein coupled receptors, what it triggers is a change in the conformation of the G protein coupled receptor so that it can now bind to heterotrimeric G proteins and catalyze the release of the guanosine diphosphate from the alpha subunit and the binding of guanosine triphosphate to the alpha subunit. And when guanosine triphosphate binds to the alpha subunit, it changes in conformation. It breaks off from the beta-gamma complex, and then both the beta-gamma complex and the alpha subunit with GTP bound, they both break away from the G protein coupled receptor, and they both then go off and have signaling roles. Okay, so if I just show this, here we now have the plasma membrane. Now, here is our alpha subunit, here, okay, and it will now have guanosine triphosphate bound to it here, so it will be in the on state, and it can go off and have signaling roles. I'll just colour it in red here, like so. And then we'll also have the beta-gamma complex over here, which will also now be on its own. Okay, and it can also go off and have signalling roles. Now, obviously, usually we don't really care about the beta-gamma complex. It's usually the alpha subunit that's more important. Okay, but it's worth noting that the beta-gamma complex can, in some cases, be important and have important signalling roles. Okay, right, now, there are loads and loads of different alpha subunits and beta subunits and gamma subunits that you can use to build heterotrimeric G proteins. A huge number of them. There's over 20 different alpha subunits when you include all the different splice variants. There are 16 known genes for alpha subunits, but because they all have multiple splice variants, there's loads of different alpha subunits. Okay, there's 6 known different beta subunits coded by 5 genes. Again, one of the genes has 2 known splice variants. And then there are 12 genes for gamma subunits, and then just 12 subunits because splice variants don't seem to be a big thing for the gamma subunits. Okay, but the point is there that there is a huge amount of diversity for heterotrimeric G proteins. Okay, and different G protein coupled receptors will activate different heterotrimeric G proteins, i.e. not all G protein coupled receptors will activate every last heterotrimeric G protein you could possibly come up with. Okay, they have their favourites. Okay. Now, the G protein coupled receptor looks at all three subunits to decide whether it wants to bind and activate the heterotrimeric G protein. Okay, so it is important which beta and gamma subunit you have, however it's not that important compared to the alpha subunit. The main thing that determines whether the G protein coupled receptor will bind the heterotrimeric G protein or not is which alpha subunit you have there. The beta and the gamma subunit do make a difference. It's not strictly correct to say that they don't, but the difference is kind of negligible compared to the alpha subunit, okay? So you can forget it and get away with it, okay? So, basically, these G protein coupled receptors, H1 and the cysteine leukotriene receptor 1, they have favourite heterotrimeric G proteins, okay? And the heterotrimeric G proteins they activate are ones where the alpha subunit is either alpha Q or it's the less famous but very similar in structure, alpha 11. Okay, so alpha Q is very, very famous alpha subunit for heterotrimeric G proteins to have. However, there is this other one called alpha 11, okay, which is also commonly found in cells. Okay, and this is basically an identical structure to alpha Q and can do basically the exact same thing, but it is coded for by a different gene. Okay, now as I say, the beta and the gamma subunits do make a difference, but we're going to forget that difference as a helpful simplification. Okay, so we're not going to fuss about what beta and what gamma subunit you have there. Okay, so we're just going to ignore that. We're just going to call those beta and gamma subunits. Now, heterotrimeric G proteins are named according to which alpha subunit you have. So these heterotrimeric G proteins would be called GQ/11 heterotrimeric G proteins, which simply tells you that they're heterotrimeric G proteins where the alpha subunit is either alpha Q or it's alpha 11. Okay, so quite simply, when histamine binds to the H1 receptor on the surface of the bronchial smooth muscle cells, and when cysteine leukotrienes bind to the type 1 cysteine leukotriene receptor on the surface of bronchial smooth muscle cells, those receptors change conformation so that they are now in a conformation intracellularly where they can bind to GQ/11 heterotrimeric G proteins. Okay, which are these heterotrimeric G proteins where the alpha subunit is either alpha Q or alpha 11. When the heterotrimeric G protein binds, the GDP is released and a GDP molecule comes in and replaces that GDP. The GDP causes a conformational change in the alpha Q/11, which causes it to break away from the beta-gamma complex, and both of these complexes now dissociate from the G protein coupled receptor, so the beta-gamma complex dissociates from the G protein coupled receptor, and the alpha Q/11 subunit dissociates from the G protein coupled receptor. And you now have these two free signaling molecules that are attached to the underside of the phospholipid bilayer. Okay, right, we're going to throw the beta-gamma complex aside, we don't care. We're going to just look at the alpha Q/11 and what it does inside these bronchial smooth muscle cells. So basically, the alpha Q/11 in the on state is now capable of binding to and activating enzymes called phospholipase C beta enzymes. So we have seen phospholipase C enzymes before in this video. However, the ones that we saw before were phospholipase C gamma enzymes. And remember, those were activated by being recruited by phosphotyrosine residues. They had SH2 domains in, which could bind to phosphotyrosine residues. We are now going to talk about the phospholipase C beta family of enzymes, okay, which is the more famous phospholipase C family. Okay, normally when people talk about phospholipase C and then don't clarify which family they mean the phospholipase C beta family. This is the family that's going to be activated by alpha Q/11 subunits of heterotrimeric G proteins. Now, there are actually 4 different members of this family. There is phospholipase C beta 1, there is phospholipase C beta 3, there is phospholipase C beta 4, okay? And we missed out 2, did I? And the phospholipase C beta 1, 2, 3, and 4. Okay, right. Now, these enzymes can all be activated by the alpha Q/11 subunit in the on state, i.e. with GTP bound. So let me show this now. Okay, so let's have the plasma membrane here. Okay, then I'll put my alpha Q/11 subunit here. Okay, and it's still attached to the underside of the plasma membrane by a lipid moiety there. Okay, so here is the GTP bound here. Okay, and this is now alpha Q/11. So let's colour this in red. Okay, so what this is now going to do is it's going to activate phospholipase C-beta enzymes. Now, phospholipase C-beta enzymes are quite mobile enzymes. They can either be soluble in the cytoplasm or they can be attached to the underside of the phospholipid bilayer. So let me show this. So basically, these phospholipase C-beta enzymes, they have means to attach themselves to the underside of the phospholipid bilayer, like so. This doesn't activate them, however. All it does is it recruits them to the underside of the phospholipid bilayer. In order to be activated, they need the alpha Q/11 subunit to bind to them. Okay. However, as I say, they're not always attached to the underside of the phospholipid bilayer. They can spend some time in the cytoplasm. Okay, so there's an equilibrium basically between phospholipase C-beta enzymes moving around in the cytoplasm, then ending up attaching into the onto the underside of the phospholipid bilayer, staying there for a while, and then moving back into the cytoplasm. Okay, so they have many different subcellular localizations basically. Okay, now the alpha Q/11 subunit in the on state is only capable of activating those phospholipase C-beta enzymes which are currently attached to the underside of the phospholipid bilayer. Okay, now as I say, there's an equilibrium between phospholipase C-beta enzymes which are attached to the underside of the phospholipid bilayer and those that are in the cytoplasm. So at any one time you will have a certain fraction of your phospholipase C-beta enzymes attached to the underside of the phospholipid bilayer. It's this fraction that are amenable to being activated by the alpha Q/11 subunit. So it will now bind to these phospholipase C-beta enzymes that are attached to the underside of the phospholipid bilayer, okay, and activate them. So now the phospholipase C-beta enzyme is activated. And then what it will do is exactly what those phospholipase C-gamma enzymes did once they have been activated, okay. It will break down phosphatidylinositol 4,5-bisphosphate molecules in the cell membrane into IP3 and DAG. So I'll just remind you of this reaction. So here is a phosphatidylinositol 4,5-bisphosphate molecule, here a PIP2 molecule, and I'll just colour it in now. So here are the phosphate groups in vivid purple there. Here in blue is the inositol ring. Okay, here in orange are the long-chain carboxylic acids, and here in green is the glycerol molecule. Okay, now note, this reaction is the exact same reaction that we saw earlier, but of course it's occurring in a different cell. It's now occurring in the smooth muscle cells rather than in the mast cells. Okay, right, so phospholipase C-beta again will cut here, and then it will produce IP3 molecules, which will now drop into the cytoplasm and go and work on IP3 receptors which are in the ER membrane. So this is the phospholipid bilayer of the ER membrane represented here. And here is our IP3 receptor calcium channel here. Okay, the IP3 will cause the IP3 receptor to open, and you'll now get the release of calcium from the intracellular store into the cytoplasm of the cell. Okay, so the question now is, why does rising intracellular calcium inside the smooth muscle cell actually cause the smooth muscle cell to contract? Okay, so to understand this, we need to know something about how smooth muscle cells are activated to contract. Now, smooth muscle cell contraction is initiated in a very different way to the way that skeletal muscle cell contraction or cardiac muscle cell contraction is initiated. So let me show you how skeletal muscle cells contract. Okay, so firstly, a little bit of structural science about the smooth muscle cells. So, Basically, smooth muscle cells are not nearly as ordered as skeletal muscle cells or cardiac muscle cells. Okay, this is why they do not have the striations that skeletal muscle cells and cardiac muscle cells have, and why instead they appear smooth. Okay, instead what you do have is actin filaments that are oriented along the axis of the smooth muscle cell, and these are attached to organizing centers known as dense bodies. Okay, so let me just put a few of these in. Now, some of these dense bodies will be attached to the cell membrane, like so. Okay, so that'll probably be enough. So these are representing dense bodies, and these are places where actin filaments, the thin filaments of muscle contraction, can be attached to. Okay, so I'll colour in these dense bodies here in turquoise. Okay, so attached to these dense bodies then, you have actin filaments, which I'll just show as these thin filaments like so, and these will be oriented along the axis of the cell. Okay, so along this long axis of the cell. Okay, so pointing in this sort of direction, horizontally. Okay, like so. So these are the thin filaments of muscle contraction, like so, and all of these dense bodies will have a few of these. Okay, right. Now, in between thin filaments that are attached to different dense bodies then, you will then have thick filaments, myosin filaments, okay, and I'll represent these by these orange lines. Okay, so these are our thick filaments. Where can I put another one? Maybe there. Okay, and basically then, the way that the smooth muscle cell can contract is if you activate these thick filaments to climb up the thin filaments, what will happen is this thick filament here, it will climb up this way, up these thin filaments, and pull this dense body this way, okay, by pulling these thin filaments in this direction. And then on this other side over here, these thick filaments here, they will pull these thin filaments in the opposite direction, and hence you'll pull the two dense bodies together. And if you can imagine this happening all over the place between all the different dense bodies, and you can imagine that the entire smooth muscle cell is going to contract down. Okay, so we've seen two key things here. We've seen actin filaments, which are these thin filaments attached to the dense bodies. Okay, and we've also seen myosin filaments, which are the thick filaments in orange, okay, which are involved in pulling the thin filaments towards the center of the thick filaments, so towards these middle lines here, okay, and therefore bringing the two dense bodies closer together. Okay, so now let's study the structure of the actin filaments and the myosin filaments in a little bit more detail so we can understand this, and specifically so that we can understand what it is that begins this process of contraction. What is it that activates the myosin filaments to actually start working on the actin filaments, okay, and pulling them like this to pull the dense bodies closer together. Okay, so we'll start by discussing what an actin filament is. So actin filaments are made from actin monomers, okay, that you can polymerize together, okay, and you have two actin polymers intertwined with one another basically to make the full actin filament. Okay, so let me show this. So here is one of these actin polymers that's going to make the actin filament, and as I say, there's going to be two of these intertwined with one another, and basically it's going to loop round in an alpha helical way, like so. Okay, so these are loads and loads of actin monomers polymerized with one another, like so, and basically they're going to be coiling around in what's known as a left-handed alpha helix. Okay, let me just explain what a left-handed alpha helix is. So firstly, what is an alpha helix? Well, an alpha helix is just a spring shape, okay? So, like so, basically. This is an alpha helix. Okay, however, there are two different types of alpha helices. There are what are known as left-handed alpha helices. Okay, and I'll try and squeeze in that. And there are also what are known as right-handed alpha helices. I just want to explain to you what the difference between a left-handed and a right-handed alpha helix is. Okay, so this alpha helix that I've got here, this is meant to be a left-handed alpha helix. This portion here is meant to be out at the front of the page, and then this is supposed to be going back into the back of the page, coming out to the front of the page, round to the back of the page. Now, why is that a left-handed alpha helix? How do I know that that's a left-handed alpha helix? Well, basically, if you take your left hand, okay, put your finger in the way that the put your thumb in the direction that the alpha helix is going up, like I have now, and then put your fingers coiling around like this, and basically the coil goes around in the same direction as your fingers, okay? So your thumb is going up in the direction that the helix is going, okay, and you can see that this coil is coiling around like that in this clockwise direction, which is how my fingers are going, okay? That's not like my right hand. If I now take my right hand here and put my thumb in the direction that the alpha helix is going, my fingers are now going in the opposite direction to the way the alpha helix goes, okay? If I went round in the direction my fingers want me to go, which is anti-clockwise, I would actually go down the alpha helix, okay, rather than up. So this is a left-handed alpha helix. A right-handed alpha helix then is the exact opposite one, okay? The exact mirror image, basically. So now this is at the back of the page, this is coming out towards the front, this is going to the back, this is out towards the front, going to the back, out to the front, going to the back, out to the front, back, front, back, front. That now is a right-handed alpha helix, and if I take my right hand and put my thumb in the direction that the helix is going, and I go round in the direction my fingers want me to go, then I'll actually be going upwards, basically. Okay, so that's what the difference between a left-handed alpha helix and a right-handed alpha helix is. Now, actin filaments are made out of two polymers of actin monomers, and these little blobs are actin monomers, and both of these actin polymers are going to be left-handed alpha helices that are going to be intertwined with one another. Okay, so this is supposed to be a left-handed alpha helix. If I take my finger here, I put my thumb in the, if I take my left hand here and put my thumb in the direction that the helix is going, then it should be coiling around in this way here. Okay, so this should be at the back basically, and this should be at the front here. Okay, now basically I'm going to put another one of these that's going to intertwine with it. Okay, and it's also going to be a left-handed alpha helix, and it will go over the top of that one. Okay, then it will go underneath this one here. Okay, and then it will go over the top of this one, so it will help if I colour this in. Okay, so basically here is this second one that I've just added in. It's going round the back there, and it's going over the top there. Okay, and here's our first one then now, in turquoise here. It's going round the back there and over the top there. Maybe that now hopefully gets the point across here, that you've got these two left-handed alpha helices, like that, intertwining with one another, and that's what makes an actin filament. So this is what an actin filament is, and each one of those little blobs is representing an actin monomer, basically. Okay, right. So that's the basic concept of what an actin filament is, and that's what those little lines that I've got that aren't coloured in are. Okay, they're the thin filaments. Now let's turn our attention to the myosin filaments. Okay, so let's start off by looking at the actual myosin molecules out of which we're going to build a myosin filament, and then we'll look at how they can be assembled together to make a myosin filament. Excuse me. Right, okay, so myosin molecules then are actually made out of 6 separate proteins that are all joined together. Okay, you have 2 what are known as myosin heavy chains and 4 myosin light chains. Okay, and the myosin light chains are going to be very important for us to know what they are. Okay, but we're going to start off by showing the myosin heavy chains because they're the more impressive ones to look at. Okay, so basically, again, the myosin heavy chains, they have these heads here, and then they too have alpha helices, and I don't know whether these are left or right alpha helices, but they have alpha helices that are going to be intertwined with one another, okay, like so. Okay, and here is this other myosin heavy chain here, which will also have this alpha helix, which will be entwined with this other alpha helix of the other myosin heavy chain here. So basically, we've got here a myosin heavy chain dimer. Okay, so these are MHC dimers. So MHC here stands for myosin heavy chain. Okay, not major histocompatibility complex. Okay, so myosin heavy chain. Okay, right. So let me show you the different portions of each one of these myosin heavy chains. So here is the head region of the myosin heavy chain. This is one of the myosin heavy chain's heads, and this is the other myosin heavy chain's heads. Then you have this first portion here, which is made up of these alpha helices intertwined like this, and this is what's known as the hinge region of the myosin molecule. Okay. Then this portion down here, this is known as the tail region of the myosin molecule. Okay, so the main bulk of the myosin molecule then is made by having these two myosin heavy chains dimerized together in this way. Okay, now attached to these two myosin heavy chains that are dimerized together, you're going to have 4 additional proteins which are known as the myosin light chains. Okay, now there are 2 different types of myosin light chain. There is what's known as the regulatory myosin light chain, which is abbreviated down to the RMLC, so this stands for the regulatory, and then it's myosin light chain, MLC. Okay, so just substitute in heavy with L to get that. Okay, and then you also have another myosin light chain known as the alkali myosin light chain. Okay, which is abbreviated as the AMLC. So this is the alkali MLC. Okay, right. So I want to now show these regulatory myosin light chains and these alkali myosin light chains attached to our myosin heavy chain dimer. Okay, so basically each one of the heads of the myosin molecule is going to get a regulatory myosin light chain attached and a alkali myosin light chain attached. So here, these can represent the regulatory myosin light chains. Okay, and each one of those heads has a regulatory myosin light chain, and then these ones, these will represent the alkali myosin light chains. So each one of the heads of the myosin heavy chains gets 2 proteins further attached to it, 1 regulatory myosin light chain and 1 alkali myosin light chain. And that produces us a complex overall of 6 proteins, 2 myosin heavy chains and 4 myosin light chains, of which 2 of those myosin light chains are regulatory myosin light chains and 2 of them are alkali myosin light chains. Now, usually myosin molecules are just schematically shown like so. Okay, this represents the hinge region, this the tail region, and this the region where the 2 heads are. Okay, why have we gone into the structure of a myosin molecule in so much detail? Because I want you to understand what the regulatory myosin light chain is, because that's going to be really important in turning on the contraction within these smooth muscle cells, and we'll come back to that in a moment. So let me firstly just outline to you now the structure of a myosin filament, because this at present, this is just the structure of a single myosin molecule. Okay, we now want to show how you actually build these orange filaments that are amongst the thin filaments here, and which are going to contract and move up the thin filaments, okay, to bring the 2 dense bodies closer together. Okay, so let me now show you a myosin filament. So basically, to build a myosin filament, you're going to put together loads of myosin molecules, okay, like so. So let me show this. So here is one myosin molecule. What you can then do is aggregate together lots of the tail regions of the myosin molecules, like so, and have the hinge regions and the head regions coming out from this place where you've got all of the tails aggregated together. Okay, so this is how you can build a myosin filament, basically. Okay, by aggregating together loads of these myosin molecules and then having the hinge regions and the head regions sticking out. So all of these tail regions of the myosin molecules get aggregated together, and then from the sides of that fibre, you then have the hinge regions and the head regions coming out from that. The other important thing to say about myosin filaments is that they are different at different sides, basically. Okay, they have opposite polarity at different sides, and this is necessary because on this side we want the myosin filament here to pull these actin filaments in this direction, okay, that direction going that way. But on this side, we want these myosin filaments, the same myosin filaments, to pull these actin molecules, actin filaments, in the opposite direction. Okay, so basically, the myosin filament has opposite polarity on different sides. Okay, so on this side, you're going to have the myosin filament polarized like this. So what I've shown you here is actually how the myosin filament would appear on this side here. Okay, but on the opposite side, what you'll have is it appearing like so. The heads will be oriented in the opposite way, okay, like so. So again, you'll have the tail regions all aggregated together to make a fibre like so, and then the hinge regions and head regions poking out from the sides. But now the hinge regions and the head regions are oriented in the opposite direction. And basically these head regions are the portions which can interact with actin filaments, okay? And they will pull actin filaments in the direction towards their tails. So when the head regions interact with the actin filaments, they will pull the actin filaments in the direction towards their tails, so in this sort of a direction here. So a myosin filament polarized like so will pull an actin filament in this direction, whereas a myosin filament polarized like this will pull an actin filament in that direction. Okay, right. So we'll call it there for this video, and in the next video what we'll do is we'll put all of this together and see how that calcium signal that we've generated within our smooth muscle cell because of the histamine or the cystenyl leukotrienes is actually going to cause the commencement of contraction.